afternoon, everyone. Happy St. Patrick's Day and welcome to our CCOVI lecture. It's my pleasure to introduce our speaker today, Dr. Belinda Kemp, our very own enologist, senior scientist here in CCOVI. Belinda joined our team just last summer, but she came to us from Plumpton College in the UK, and prior to being there for a couple of years, she completed her PhD at Lincoln University in New Zealand, where she was doing a lot of research on Pinot Noir, and has spoken to us at the Fruit and Veggie Conference a few weeks ago now on that topic. And before she completed that stint, just after she completed that stint, she also worked as a commercial winemaker in the UK for Nye Timber, and that's where she also gained a lot of experience in sparkling wine production, and that's another research focus area that she certainly has expertise in. Her position here at CCOVI is quite unique because half of her time is spent on applied research to topics directed by the industry, but the other half of her time is spent on knowledge transfer back to the industry. So ensuring that all the great initiatives that we start here in CCOVI and all the research outputs, we're able to put back into practice within the industry. So currently her research focus is on sparkling wine and also tannin management in red wine, and today she's going to talk to us about her new program on tannin management that has been coined Tannin Alert. So we're really looking forward to hearing her talk today, and I'll leave the floor to you.
Thank you. Thank you, Debbie. Thank you. So just to begin with, just to go over, I'm going to talk to you today about ripeness definition with regards to what it means to the winemaker, but also to the grape grower. Tannin structures, just briefly. I'm not going to enter into a chemistry lecture. You get enough of that, and somebody else can do that for you. I'm going to talk to you about tannin management and what that actually means. About skin tannin variation as well, about vintage variation, seed colour, very important and a good indicator as well. What the plans are for Tannin Alert as an outreach programme, where we are at the moment and what the next steps are. And we're going to talk about classification of tannin, of our grape varieties that are used in the programme by tannin concentration. I'm just going to briefly mention some tannin management options for red wines for the Niagara region as well, which some people are starting to explore at the moment, or have been as well. So, to start off with, just a reminder, we know that during the pre-harvest monitoring programme, Jim and I nip out, well this year, before that was Jim obviously, and his colleagues, but we nip out to the vineyards, 4 sites, and we're looking at sugar levels, and we do them here by Brix, pH and acid. But also one of the really important and more common ways of determining ripeness as well is phenolic compounds and aroma ripeness. Well, what do they actually mean? Well, first of all, when we talk about phenolic ripeness, there's no such a thing as phenolic ripeness. Let's just put that in the clear. What we're really looking at is the type of tannins or the type of anthocyanins that we actually want as winemakers in our wine. So we can get rid of the ones, or not get rid of them, but manage them during winemaking. Can't get rid of them, but we can manage them a bit better to make the wine style that we particularly want to make using that variety. So we know from here in Niagara, from a lot of the work, that the late Mr. Andy Reynolds, that sounds awful, but he arrived late is what I mean. And his work looked at, through many vineyards in Niagara, looked at light and temperature, but more importantly crop load, vine water status, soil and vintage differences. And Andy's had a look a lot at total phenolics, anthocyanins, those sort of things, and also flavonthiols, which are the smaller tannins. And his lab has done an awful lot of work in that area over the last few years. So just a really quick introduction to it. Phenolic and aroma ripeness. One of the things, can I move? Yeah. One of the things we're looking at here and do a lot of work on is, of course, methoxypyrazines, and C6 compounds are now getting an awful lot of coverage and research being done in Australia on it at the moment, but also in Texas, Argentina. A paper was just released from Argentina, Washington State. So a lot of people are investigating the ratios and the combinations of the C6 alcohols and aldehydes at the moment. So basically what this graph is supposed to be depicting is the green arrows head towards green aromas or flavours, and the pink ones are to represent ripeness, if you like, of flavours and aromas. So our flavonthreols over here are catechin. I've used in this example catechin and epicatechin gallate. Now I'm going to talk a bit more about the differences between skins and seeds, but epicatechin gallate is predominantly in the seeds, and it's been shown in recent years that this particular flavonthreol can contribute the coarseness and the harshness and the roughness within a wine. So for instance, if you did extended maceration and you had slightly unripe grapes, then you would be extracting more of these guys, more of the the coarser tannins. The proanthocyanidins, I'm sure you all know, but they come predominantly from skins. They're longer chained condensed tannins within the skins, whereas the seed ones are much, much shorter and often easier to extract towards the end to ferment, I guess is what I was trying to explain. The other compound that's really of interest to us as well, or compounds, are the C13 noisebonoids. So this is beta-damascenone. In the work I did in New Zealand, we found that with Pinot Noir, when they're exposed to light, beta-damascenone levels increase dramatically, and they're really important, this compound, because it contributes it contributes to the fruity flavours, but it masks any green flavours as well. But in a recent paper just released last week or the week before from Tasmanian group who worked on Pinot Noir with regards to vine vigour, they found that in Tasmania beta-ionine increased in the low vigour vines and was much higher in Pinot Noir than in New Zealand. But both of them have similar, or thought to have similar impact on the aromas of a wine. So we're really looking for less of these green compounds, less of the flavonthreols in a wine. We want more of the skin tannins, and we want more of those fruity aromas. And that's basically what people are talking about, winemakers are talking about when we talk about ripeness, phenolic ripeness, and aroma ripeness. So in our seeds, we have much higher flavonthreols. We have much higher levels of this in the skins of the longer chained, higher level proanthocyanidins, which are much smoother when they're extracted into the wine. So one of the things we're trying to reduce is over-extraction of seed tannins, and this can sometimes happen in poor years, or difficult years, I should say. And this over-extraction of seed tannins combined with these sort of green aromas that can sometimes happen that haven't managed to decrease at all at harvest time can sometimes give rise to the term green tannins or unripe tannins that wine writers and winemakers sometimes use, the greenness. Although there's no actual green tannin, because tannin is colourless. It's just this perception of it with the aromas there. And of course we want those higher skin tannins. So that's just a quick background for you. So people often talk about these days integrated tannin management, which is quite simply a combination of the vineyard influence and what we can do in the vineyard, and some things we can't do at all. It's completely and utterly outside of our range with regards to weather and climate, for instance. And of course what we can influence though is when the grapes come into the winery, and the winemaking influence we can have to reduce that greenness, to reduce the harshness, and do things like alter the temperature of the fermentations, keep an eye on the ethanol levels, which we would do before anyway in the vineyard when we know what the sugar levels are. But there's all things we can do within in the winery that doesn't necessarily cost loads of money with regards to equipment. So, just to recap, immature seeds, these green seeds contain the harsh phenols, which can be extracted during ferment, but more often towards the end of ferment. Skin tannins are released, skins release their tannins early on in ferment, whereas the seeds come out at the end. So what we really are after are the tactile skin tannins before they become overripe. Okay, so that's really what we're looking for in our grapes. So just briefly, because it really isn't going to be a chemistry lesson because I've got other things to talk about. But these are just an example of some of the flavonthiols. Here's catechin, its isomer epicatechin, and epigallocatechin, which is mainly and predominantly found in the skins. It's one of the ways we can tell the difference between, in a wine, what are coming from skins and what are coming from seeds. Is this with the gallo portion in the middle of there, epigallocatechin, whereas epicatechin gallate comes from seeds. Okay? So, further within that, we've got two other subclasses. These are pretty cool compounds as well. And we have procyanidines, which are found in skins and seeds, but predominantly in seeds more than anything. And there again is where you find the epicatechin gallate. Then our prodelphinidins, which are found only in the skins, which derive from epigallo or gallocatechin as well. These all polymerize as well, of course, to make longer chain compounds. However, one of the important things is that you won't get anthocyanins polymerizing with tannins in the grapes, so you won't get colored compounds or colour pigments within the grapes. That only happens when it gets into the wine. So if you have a nice combination of the both, where they combine and blend together, it gives you stability, aging potential, stronger colour, etc. Okay? So tannins are colourless. Tannin synthesis is influenced by UV light more than temperature, anthocyanins by temperature, and particularly in the UVB range. So light more than anything. There'll be differences in seasons, there'll be differences in regions, there's differences between grape varieties, there's differences between grapes on a bunch, bunches on a vine, vines in a vineyard, vine vigor, etc. So you can see that trying to pin it down and to investigate it in detail can be difficult. However, there has been quite a lot of work done in this around the world, and one of the things I wanted to share with you was that in Australia, in McLaren Vale, they looked at Shiraz grapes there, and they wanted to have a look and see what was happening tannin-wise within the vineyard, and they found that high tannin levels at flowering were happening. They had high tannin synthesis towards veraison, and then from then on it declined until harvest date. However, in another area within Australia, in the Sunraysia, northwest of Victoria, a different region completely, they looked at Shiraz and Cabernet Sauvignon skins. And what they found there was that the tannin levels peaked at fruit set, okay, but then declined from then onwards. So whatever they had at fruit set, that was it. Nothing else was happening. So very, very different in different areas. This was a study done by Mark Downey from the AWRI with many, many samples, by the way, over quite a few, I think it was 3 or 5 years. I'd need to check. So what do we know in the vineyard here in Niagara about tannin synthesis within our region? Well, from some of the work that Andy's done with regards to phenolics and flavonthiols, we can see that many things, like I mentioned, will have an effect. Vigour, site, variety, but we haven't pinpointed or got a database to show categorically what is happening with regards to skin tannins and what's happening with seed tannins, although that might not be so different as skin tannins will be. And that's where, partly that is where this program of Tannin Alert came from. But also equally, when I arrived talking to winemakers and talking red wines and things, it's one aspect that they really wanted to investigate more with regards to Niagara region and Niagara varieties that are grown here. So tannin, people talk about tannin quality. And that's really, again, goes back to the degree of polymerization or the length of those tannins, and also its association with other molecules within the wine as well. Now, they combined with, the catechins in particular can bind with so many other compounds. The other thing to remember through all of this, though, is whatever winemaking technique used, some tannins will always remain in the skins and seeds. There's always going to be that. Now that could be from yeast, it could be from anything you do during the wine making production, but equally it's the binding within the cells, the interest, those tannins staying within the cells, locked in there. So we can't take everything out, and nor would we probably want to. But we can get an indication from the grapes, skins and seeds, of whether we're going to have a low, medium or high level within the wine. Okay. So Bruce Zockline's lab in Virginia as well also found a relationship between grape maturity with regards to Brix and TA, total seed phenolics, but also within that seed tannins in their Cabernet Sauvignon. So they've done a lot of work in Virginia on this area as well. In fact, there are many regions in the world of wine that aren't investigating tannins on some level on a regional and sub-regional basis. And we are too now. So one of the difficult things is we don't know what the ideal grape tannin content is for any wine or any given wine anywhere in the world. It's going to be different from year to year, it's different for wine style, etc. So that can be a challenge for researchers, but it's also a challenge for the winemakers as well. The other thing is there's other factors within the wine that will affect your tannin perception. So one person obviously is going to taste things differently to another. So what one person will think is quite harsh or rough will be described in another way or felt in another way by somebody else. That's due to their saliva makeup, not to anything what you've eaten beforehand or whatever the case is. But other factors affect the wine. So glycerol, acidity is a top one, manoproteins. Again, going back to the source and the structure, that difference between the skins and the seeds. Whether the anthocyanins and how much you've got of those in there. Pectin, ethanol levels make a huge difference, the heat of the wine. Sugar and individual taste, which I just mentioned. So the amount of tannin in grapes is significantly higher than what we get in the resulting wine. All right? And that's also through extraction purposes and methods that we use for extraction, which I'll show you as well. So this is a graph from Australia from some of their studies that they've been doing, and this large arrow which I pointed down to here is basically Pinot Noir. And what this graph is showing is the amount of milligrams per gram of fresh weight and is total tannin concentration within the skins of these varieties. So Pinot Noir is there. You see Nebbiolo is really low, interestingly enough, but over here we've got two different types of Cabernet Sauvignon, much higher there, so you can see the difference between Pinot Noir and Cabernet Sauvignon. So this was done over several years on skin tannins in different regions of Australia. So it's going to be very different for us. One of the things that's interesting as well, I'll just pop in there, is most of the varieties with a very high tannin levels are actually table grapes, surprisingly enough. Whereas our wine grapes seem to be, overall, across the whole graph, seem to be much lower. But from there, we've also got a Merlot, which is sitting here, just higher than Pinot Noir, but not quite as high as Cabernet Sauvignon. That is across several years and across several grape varieties. So we have got, as I said, variation across the whole vineyard and blocks, etc. There's a significant variation in skin tannin can happen within the same vineyard. And one of the things that Mark Downey in Australia came up with from his research is that it's associated very much with vine vigour. But that can also, as the work that Andy said done, could also be down to things like crop load and other aspects, and vines' water status, as we said. So it's important to note as well that from 1 year, so from last year, whatever we had in the we're about to find out as we go to put the analysis through the well plate reader. From 1 year to the next, you're going to have very different tannin levels and tannin concentrations, particularly in skins. Seeds don't tend to change quite that much unless something shows up in our varieties here while we're monitoring. It'll be quite interesting. But generally it's been found that seed tannins don't really change as much during ripening other than the colour. Well, you'll see in a second, I'll show you. So, taking on board the ripeness, the CCOVI pre-harvest ripening program, this is actually Cabernet Sauvignon, I'm sorry, it's not at all actually, it's the Brix levels for Cabernet Franc from Site 1, and this is over the 4 years it's been running. So you can see a difference between the ripening stages between vintages. So this is just a snapshot. This is just the sugar side of it. Now I want to show you, this is a really cool picture because this is Site 3. And this shows a really clear, distinct difference between years for sugars, and one is going to expect that it won't be the same. It obviously won't be on those levels, but we would expect the same to happen with tannin concentrations per year as well. Tannin concentrations don't coincide with sugar and acids by any stretch of the imagination, and I'll show you that in a second. But what a fantastic graph to show vintage differences with regards to ripeness. And this one is for Cabernet Sauvignon, and this is Brix comparing all four sites actually that we covered this year. And you can see again the differences not only in ripening, and it would be really interesting to see when we have the data to compare and have a look at the tannin concentrations at these points as well. So hopefully we'll be able to do that when we've finished all the analysis. So this is something we did during pre-harvest monitoring, and this was purely to show the ripeness of the seeds as we're going along during ripening as we're testing for sugar levels and acid levels and pH levels. We started at 15 Brix. Now we know that nobody's going to pick at that level, but it was more to show you the seeds and what's happening within the seeds, and you can follow those along during Brix level. What's particularly interesting is the difference between 22.3 Brix and the colour of the seeds at 22.5 Brix. So even though that's a week later, because we were doing weekly sampling, that's a big difference in colour, and these seeds were randomly selected from each of the samples as we were going along testing. Now, there is a way that Patrick Island, Ristic and Island, put together to look at seed colour ripeness, and I can easily pass that paper on to anybody who would be interested in that. But it's a way, as you can see from the slides earlier from the seeds, it's a way of looking at the ripeness or getting an indicator of seed ripeness of your sample. So that's one way. You need people available to do it. It's a lot of counting of seeds, and it depends how large your vineyard is to be able to do that, and how many people you can spare to do that. So there's a lot of, it's accurate, it's useful, but you've got a lot of work hours, labour hours to be able to do it. In their study, they look at the ventral side of the seeds and the dorsal side of the seeds. I was going to say fin, but a side. And then basically are looking at taking a percentage of how many are green. But you've got to be trained to know what you're looking for with regards to green, and what percent. Is it the whole seed? Is it one seed? Is it a bit showing? So it gets a little bit finicky. However, the picture on the left, these are seeds ripening, thanks to Mary putting them under the microscope with me. And you'll see the seed in the far corner with the skin drying up. Well, that's actually what happens as the seeds are maturing. But the tannins that were in there actually do go and bind into the layer below. That's some work that Mark Downey published again as well. So the seeds, the tannins are moved elsewhere within there as that starts cracking. One of the things to remember as well, actually I'll tell you about that in a second and explain. So the AWRI in Australia provided, as you could see from the graph earlier, a big project surveying many Australian vineyards to get a database and be able to benchmark varieties, skins and seeds from across the country in different regions. So they have a web-based tool called WineCloud, and they basically use it to monitor grape maturity. You can actually track the wine ferments going along and see what's happening with regards to tannins within the ferments. You can then follow them during aging if you're a winemaker out there, and basically it helps winemakers there to understand what's happening at each stage of the process. So Tannin Alert is kind of based on that. I'll be fair, we did, I have slightly pinched some of their ideas, or borrowed them, I should say, but not the whole thing. And it's what we're doing with Tannin Alert is specific to Niagara, is different, and we're just at a starting point. We're not actually going to this to be able to provide this at this stage, but it's a starting point for our region. So what are our aims and objectives of Tannin Alert? Well, the first one is, as I said, I need to reiterate, we're developing a Niagara-specific regional database for future benchmarking and grouping of tannin concentration in the 3 main red varieties. Okay. The second thing is that it aims to provide winemakers with this information, the concentrations at harvest of these, which will then assist in management decisions during the winemaking process. And many times, most of the time, basically we have to deal in the winery with what nature has given us in the vineyard. And you could have done nothing else. It could be a really difficult year, and it's working with what you've got within the winery. So it's for the winemakers, okay? So basically we are looking to also, like the Australians, group our tannin levels in groups of low, medium and high tannin. Now at the moment we don't know what that's going to be, because we haven't got total tannin concentrations in skins and seeds done before. So we're going to work to begin with peer-reviewed papers and research done in other cool climates, so we can have a look. After the first couple of years, then we can start manoeuvring and manipulating that tannin data and concentration into our own specific Niagara groups of medium, low, medium and high. But to begin with, we need a starting point. The other thing with this is that we're looking to provide red winemakers in the Niagara region with different techniques for different tannin concentrations. Now, we're not telling anybody what to do at all. What we're doing is we're making suggestions that you could also think of this, or you could also consider that. And so basically the groups would be low, medium and high, just like the Australians did, but ours is going to be broken up as skin tannin. So you've got a low skin tannin, and you've got a high seed tannin. What would you do to get a consistent wine compared to the one last year, as much as you possibly could? That's what this is about. Okay, so this is just a little example of what the Australians came. N stands for number, so the number of samples. Cabernet Sauvignon, Merlot, Pinot Noir and Shiraz. And the low levels for Cabernet Sauvignon there started 0.1. 0.3 for Merlot, 0.4 and 0.4 for Shiraz and Pinot Noir. But you can see as you're going along, those groupings are very different for each variety. And what would be cool is we'll be able to break ours down by particular sites as well, to particular areas within the Niagara region for our 3 grape varieties. Okay, so we're going to be doing extracting the skins and seeds. Actually, we've started, to be fair. We've got Cabernet Franc from 4 ripeness sites. I say ripeness sites, ripeness monitoring sites we use this year. Cabernet Sauvignon, Pinot Noir from 2 sites, which we hope to increase a number of sites for Pinot Noir this year. But there's 3 varieties for the time being. We use a method by Fragosi in 2011 to extract our tannins, and this is because, for me, he uses a hydroalcoholic solution. He uses acid and heating in a short space of time that is more representative of the winemaking process than 70 or 80% ethanol or acetone for me. So that's what the lovely Esther has been busy helping with in the lab, and she took these pictures, by the way, Esther Onguta at the back there. And this is us before extraction, afterwards on the skins, and there's the seeds over there. So, for the actual tannin analysis, we're using the AWRI methylcellulose assay, and this is because it works for me before so well with Pinot Noir, but it also works with Cabernet Sauvignon and Cabernet Franc. So we're going to use Mercurio's method, and we've got the well plate reader in place, and we've got hundreds of samples already extracted, ready to go to have the analysis carried out. So in the first year, we're going to have a database of Niagara tannin concentrations in 3 varieties and in their skins and seeds. We're not doing whole berry analysis, and one of the reasons for that is that skin tannin decreases as you get towards harvest, or I should say extractable skin tannin decreases as you get towards harvest, and seed tannin increases. So if you do a whole berry tannin sample, if we did do that, then you wouldn't get much of a difference, and you wouldn't actually see, it wouldn't be that helpful to a winemaker. Whereas a skin and seed tannin combination gives us more information as winemakers as to what's going on. So, as I mentioned, we're going to look at the cool climate peer-reviewed work that's happened around the world for the first year to start grouping things until we have a bigger database available. We really need 3 years, 3 growing seasons worth of data, but really ideally we'd want 5 years, and we'd just keep adding to it and adding to it and adding to it. And then you could compare it to the weather and climate each year. We could also look at the ripening process each year and any other strange events, I was going to say. I'm just thinking of winter this year, that might happen from year to year. So, following the tannin analysis at commercial harvest, we'll then be making wines, still red wines, from all 3 varieties using exactly the same winemaking technique regardless of what tannin levels they come in at. Okay, so as if you would make them all the same. Then a second set of wines will also be made using different techniques tailored specifically to the tannin concentrations of skins or seeds within there. We'll be looking at doing chemical analysis on those wines and of course sensory analysis, and we'd like to do that with the Niagara winemakers later on, much further down the line, so that we can actually see, taste, feel what those wines are like using those different tannin concentrations. So just briefly, I talked about this at the Ontario Fruit and Vegetable conference the other week, but it's just a brief brief reminder that sometimes there are many ways to deal with different tannin levels and tannin within the winery. One of the ones we were talking about the other day at Niagara College was thermovinification, flash dente, and microoxygenation. The thing is that all those 3 techniques require equipment and therefore an initial outlay of money. However, other things less so. So delestage we can do, which will eliminate the tannins quite early on, or high tannins quite early on. Saguenay to reduce the skin to pulp ratio. And we could do, some people consider short cold soaking, but it very much depends again on the ripeness of the aroma ripeness in particular, whether that's any use. It's been shown to extract more colour and aromas, but not really tannins, so unless you use enzymes. So that might be something for consideration in some years, but not for others. Cap management, we all know about that. Extended maceration, well, if you've got loads of seed tannins, high seed tannins and less skin tannins, that's not necessarily something that we would want to do either, to leave it on the seeds. Another one, an easy way out would be to blend early on the high tannin wines with the lower tannin wines. The other thing is in good years, where you've got really high ripeness levels, for instance, or high tannins, you might want to consider a ferment with 30% whole berries in, or whole clusters even, or only a percentage, not the whole lot, or a combination of all. Tannin addition, not sure we always need to do that, but that depends very much on the type of tannin you're adding, the timing, and the wine style you're after as well. We can alter fermentation temperature, speed of the ferment. We can do that as well. We can press early as well. Take the wine off the skins early. The high pressure presses mean that you extract far more harsh tannins, whereas the lower pressures, I should say, means that you have less of the skin tannins, harsh seed tannins, sorry. And of course you could hold some of the pressed wine back. You don't need to blend it back in right away. You can have a look, and if you feel like you need some astringency back in there, then you could pop that back in. You could remove the free run juice altogether, put that into another tank, and of course all these things do require tank space, I do realise that. So a higher concentration or high concentration of extractable C tannins has been clearly shown to negatively affect wine quality. And it used to be that people thought, oh, we can extract the C tannins and then they'll all bind together in the wine. Oh, that's going to be fantastic. And we'll have a lovely smooth wine because we've got the base units there. That isn't necessarily what happens at all. So we need to be careful of how we handle our C tannins. But again, it comes down to wine style. So if you want to make a young red wine, it may be that you restrict the tannins a bit more and increase the pH. So there's different things that we can do for different wine styles. So what are we going to do? Well, by this time next year, hopefully, fingers crossed, we'll have made all the wines for 2014 vintage. It's just important to say that even though we've got 3 varieties on 4 sites, if we did microvinifications in triplicate for all of those, we'd end up with 72 microvinifications, which by any stretch of the imagination is impossible for one person. So we are going to work out on sites and be site specific and have a look at different varieties and try and at least get one set of microvinifications for each variety. All right, so we're working on how the best way to do that at the moment. Plus we'll have all the tannin concentrations from the skins and seeds. We've got most of them, all of them have had, most of them have been extracted, the tannin's been extracted and they're waiting for the analysis to happen, which should be in the next week or 2. And then we can also compare not only to whether if we so wish to, but more important to have a look at Brix and TA and pH levels as well. So we can compare standard parameters, ripeness parameters, with phenolic ripeness levels. So that's it for the moment. I just wanted to say a couple of things. One of the main things is that this is really only a starting point. We're in our infancy with this, and it is specific to our region, so we're taking it very slowly. But it is important that I do want to thank Esther Ongata. I've been practicing her name all weekend as well. Is that right? Kind of, okay. I know. And Esther's just, she's been so helpful in the lab with me. It's brilliant. And of course, CCOVI's financed it. If we want to go into more varieties, more sites, which would be fabulous, but we need more people, more hands, and a lot more equipment, a lot more work, and then of course that sort of thing needs a big funding program to be able to do that. So this is our starting point, if you like, to have a look and see what we've got at harvest in the winery, and then how we can best make the best possible wine we can do year in and year out, regardless of what nature gives us. Okay, and then I've got some references for anybody who'd like to read up some more. Oh. Any questions?
So we've got time for questions from the group, so please, I'm sure there's a few brewing with all the massive sample analysis and the database and the potential for where this is going to go forward.
Casey, come on, you know you want to.
Belinda, just one question. I know throughout the talk you were focusing on Niagara winemakers. And I know for practical purposes right now, for the pre-harvest program, our sampling sites are here in Niagara. But
yeah.
Of course, we're hoping this is going to extend not just in Niagara, but across other viticultural areas in Ontario.
Yeah.
And potentially down the road, is this something we could look at developing into a national initiative?
Absolutely, yes, absolutely. I mean, we need to start small to begin with to actually work out how the program's going to run, what can be included where, and the winemaking side of it as well for our region. And then other varieties, other regions outside of Ontario would be absolutely fabulous, yes, without a doubt.
Yeah, because it would be interesting to see too, just how these variations change from a region like Niagara in Ontario versus those same varieties, but in the Oenology and Columbia.
And it would be so great to include Merlot. Somebody asked me about Merlot going in there, one of the winemakers, and another winemaker asked me about Syrah. And it would be wonderful to be able to build a database with all those other red varieties that are used so commonly here as well. But as I said, it's a huge undertaking to get to that point. I think we need to set up the programme, make sure it's, check each step of it, and then we can roll it out to other areas.
I'm just wondering if there's been feedback this far from the industry. Are winemakers happy to hear about this ongoing research and this?
Yes, we've had quite a number of winemakers come up and talk to me about it, both at the conferences, at the Ontario Fruit and Vegetable one, but also when I see them in the wineries and at meetings and things like that, we talk about it. Yeah. It's where it came from originally, was from them asking about it, about tannins and about what we could do in this area.
Any other questions? Well, with that, please join me in thanking Belinda. We have a token of appreciation, keep you warm in our cool winters here with a cup of tea or chocolate. Fancy. And also just a reminder that please come back and join us next Monday, March 24th, where our speaker will be Dr. Andy Reynolds, and Andy will be speaking to us about some travels he's recently undertook as part of his sabbatical. So the topic is Mediterranean horticulture, travels in Tunisia and Portugal. So we look forward to seeing everybody back here on Monday, and for our online guests as well. Take care.